Welcome to my short poster presentation. My name is Chris Baumann and I will talk about the first evidence for synanthropic behavior in late Pleistocene ravens. Synanthropic behavior means that animals have adapted to survive in a human-made environment. We see this behavior right in our own backyards, in the cities and villages all over the world. Everywhere, pigeons, crows, foxes and rodents have adapted to live among humans. However, this behavior did not occur in the Neolithic with the first permanent houses and villages, but much earlier. Today I will talk about our study of Gravettian ravens from Moravia, which already showed adaptations to humans 30,000 years ago. Moravia is located in the east of the Czech Republic. Here we studied ravens from the archaeological sites of Pavlov, Predmošti and Dolní Věstonice. In these sites, tons of mammoth bones were found, which formed the main prey of the Gravettian people. Raven bones made up the majority of bird remains, although the bird bones were small compared to the mammoth remains. Usually, if you find cut marks, you can assume that the feathers were used, and if you find burnt bones, the animal were probably eaten. However, none of these could be found in the sites. If they were not hunted by human, why were there so many raven bones at the sites? To answer this question, we studied the diet of ravens using stable carbon, nitrogen and sulfur isotopes. We sampled raven bones, extracted the collagen and measured the isotopic values using an IRMS. Carbon and nitrogen isotopes were used to determine the diet by using mixia and reconstructed trophic niches by using SIZWG. Sulfur isotopes help us to divide ravens into three groups: ravens that were at the baseline of local herbivores, ravens that had lower sulfur values, and ravens with higher sulfur values. As a result, we could see that ravens that matched the sulfur baseline of local herbivores had a diet similar to humans. The main protein component was mammoth, while the ravens with higher sulfur values had a different diet similar to large carnivores. This led us to the following conclusion. Mammoths are known to have been the main diet of humans, both from the tons of mammoth remains from the archaeological site and, of course, from the isotopic analysis. The ravens within the local baseline had a similar diet as the humans. And same is true for the ravens with the low sulfur values. Both groups of ravens seem to live near humans long enough for their isotopic signature to adapt to the local resources. The high sulfur raven had a completely different diet. However, since they were also found at the sites, we assume that the isotopic signature had not yet changed. Collagen has a slow turnover, and the measured isotopic values reflect the average values of several years. Therefore, it is possible that these ravens were attracted by human hunting activities and a high amount of anthropogenic food waste. If this study has piqued your curiosity, I would be happy to meet you at our poster and discuss the topic with you. You are also welcome to visit my ResearchGate page to discover more about my projects on paleosynanthropism. Finally, I would like to thank my co-authors and my colleagues in the Biogeology Working Group in Tübingen.